everybody for being here, and thanks to the organizers for the nice invitation. As Peter mentioned, I did my PhD and postdoc in protein folding as a general terms. But since 2006, we started working on three-dimensional genomics, where we used all these methods for protein structure prediction, or the ideas at least, and we did try to translate them into genomes and how the genomes fold. And I'm very lucky that during the morning, since Elaine's talk this morning until, if you've been in the previous talks in this session, they've introduced many of the aspects that I'm going to be talking to you. So I'm just going to glimpse over there. My talk today is about the work of Marie Troussard. She's sitting here with help some people in the group, where she's been looking at assessing the limits. How far can we go with the data that we get into model three-dimensional genomes? So you're going to be seeing here toy models, not actual real structures of genomes. But let me first tell you how we modeled a bit, very briefly, very quick, and then we go into assessing these limits. Don't have to convince you by now that these things happen in 3D, not in linear fashion. So the enhancers need to be nearby the gene to activate, and so on and so forth. So this happens in 3D, and that's the reason why three-dimensional genomics is picking up very quickly lately. Thanks to the appearance of the chromosome conformation capture technology since 2002, and most importantly, the high-C version of it in 2009. In between them, we had 4C, 5C, and now we have many other Cs coming almost on a weekly basis nowadays. But the idea is that we do these experiments. This is a high C. Never mind exactly what the protocol is, but at the end you get this type of maps. This is genome-wide, and this is just a part of a genome, where you have a diagonal, very prominent interactions, and then if you zoom in, you start seeing what has been called topologically associating domains. And these stats seem to be co-regulated, and we've heard very nice talks this morning telling about that. What we do, computationally speaking, is quite different of what Angelo has been talking this morning, in the sense that we don't use polymer physics at all. In fact, we don't use physics at all in our models. What we do is we translate these interactions frequencies, and we use them as a proxy for distances. And when we have translated these interactions into distances, we create restraints between the points. Each point here could correspond to one being in the interaction matrix, and we put restraints between them, and then using Monte Carlo simulating annealing, we find solutions that satisfy the restraints as much as possible. They never satisfy 100% those restraints, just because it's an average picture of what happens in millions of cells. And each one of them might have a slightly different conformation. So that map here contains many different conformations. So what you do is you generate an ensemble of solutions, not just a solution. And that ensemble of solutions, we send it back to our experimental friends, that they can refine the experiment, do yet another experiment to see if we can get additional restraints to, let's say, pinpoint a bit more what's happening here, which seems a bit hairball in the solution. And we do that until we run out of money, because this is expensive, or we publish the paper, whatever comes first. So how we go into these interactions to three-dimensional models, this is not rocket science, has been done for decades in the NMR field. The NMR field, the spectroscopies never actually see the atoms in the space in coordinates, X, Y, Z. What they see is this, is a two-dimensional representation of this. And what they do is, because they know the co-resonance of the protons is proportional to the distance between the protons, they can now put distance restraints and build a three-dimensional model or an ensemble of them that satisfies all of these distances. We do the same. We have an interaction matrix. We transform those interactions into distances, and this is what we spent most of the time in the group at the beginning when we developed that together with David Bau, also sitting here. And we transform this into distances, and then we use the same trick that they used in NMR many decades ago to build an ensemble of models. A bit more details, repeating myself, but it's important. Red means proximity, 2 points will be close in the space. White means not interacting, get me apart, I don't want to see you in the models. And that means repulsion. We put in restraints, we build an ensemble of solutions, and we analyze those solutions. So we've been doing that, and not only us, many people actually around the globe have been doing that, and these are just examples from the first one in 2008 based on imaging to recent ones, and I've stopped adding here because last year has been a plethora of different models coming up. But we all face the same problem, are these models correct? Are we actually really representing a correct three-dimensional structure of the genome that we are interrogating with this 3C-based data? And the only way that we can prove that this is correct is by imaging. And every single of this paper had to go together with fish imaging to prove that they were correct. And unfortunately, fish, it has a low resolution compared to the resolution of the models that you get. So you can only partially validate. For example, here is something that we published some years ago. We had two pseudoglobules, if you wish, or globules, and we wanted to see that if that distance between the two globules had a certain probability, and we look at imaging, and we proved that that was the case in compared to inactive chromatin, where this was all together. So we can partially validate. So with Marie, and that's a paper we published in March in Nucleic Acid Research, and this is what I'm going to be talking about, we come up with a strategy to build what is called toy models. We're going to build toy models that we know what structure they have. What we want in these toy models is that they have certain properties, and when we have them, we generate interaction matrix from the models, and then we reconstruct them to see whether we recapitulate the original model or not, and how far we can go. So this was done by Ivan Junier, who is a polymer physics guy, and he did these simulations. These are the toy models. These are not the reconstructed models. So we built circular chromosomes of about 1 megabase, and we had two states. It was interesting to know whether the tile-like has an effect or not in the reconstruction of the models, so with tile-like, non-tile-like. And then we had 3 levels of variability here, which are important for when you do this high-C experiment. The first one is the resolution. We built a point in the models for 40 base pair per nanometer, 75 base pair, or 150 base pair per nanometer. So you have more points here, less points here, and we built sort of an increased resolution of the matrix. The second variability is the structural variability. So we built each one of these superimposition of models, of the toy models, have 100 models in here, 100 of them. This one, they are all very much the same, so it looks only like a single model, but you actually have 100 of them. So the interaction matrix here will be very deterministic somehow. And that one has a bit more variability, that one has a lot of variability. And we want to increase the structural variability that we use to build the models, to see whether that has an impact. And finally, the noise. When we decide that 2 particular points in these models are a contact, we play a probability game and we accept it or refuse it depending on this probability game, and we accept more or less times these interactions. So we add a bit of noise into the data. Okay, so resolution, well, TAT or not alike, resolution, structural variability, and noise. And we're going to play with these 3 things and see what affects more. Let me show you some of the examples of the interaction matrices that we get from the toy models. These are 4 of the 100 toy models that we built for this interaction matrix. You see they look very much the same, blue to red, and these are the beginning and the end of the chromosome, or that particular structure. And this is the contact mark that we get. As you increase the structural variability, this is a bit more structural variability, and this is a lot of structural variability, you see that the matrix gets a bit more sparse, more smooth. And guess what? You start seeing the TAT very clearly when you have a lot of structural variability, because it's the only thing that they have in common. These guys here, the only thing they have in common is the fact that we force TATs to occur. Okay? So the question is, now I have these interaction matrices. We built 67 of such interaction matrices with variability on the noise, structural variability, etc., at different amounts. This is one high-resolution matrix. This is a bit less resolution matrix, 600 pins, here 200 pins. Both of them have very little structural variability in this case. So this is the actual toy model. When I put that into our software called TATbit, this is what I get. I get these structures here. Now the question is, can I superimpose this to this and see how it looks like? And this is the numbers here. This is just the simulation. But you get 2 measures, distance RMSD, which goes between 0 and infinite, and is scale dependent. So it actually captures whether the scale is correct between the two models. In fact, we almost never get the scale perfectly. That needs to be said. We need to work a bit more on the scale, but this is not far from the reality. And this is 32 nanometers. These objects are really large compared to proteins, where people are used to angstroms. And then this is another one where we have a bit less structural variability. These are a bit structural variability, and these are the numbers. The second measure is this distance dispersion correlation coefficient, which is scale-less in a sense. We just correlate the distances between them. 1 means a perfect correlation between distances, 0 means imperfect correlation. So the numbers are pretty decent for these things. So let me tell you what are the effects. I mentioned that we had these 4 things that we variate. So TATs. Are TATs good or not for modeling? And in fact, they are good for modeling. And we are very lucky that most organisms have TATs because they help us to model. And this is the Hermes distance RMSD measure, a quality measure. This will be a random throw of points in the space. So if I go beyond that point, it means that my modeling is really bad. And this is the resolution, high resolution, low resolution. And in dark colors, you have TAT-like, and with light colors, you have non-TAT-like models, the ones that do not contain TATs. And don't ask me why exactly. We're still struggling to understand why. But when we have TATs, our modeling does slightly better than when you don't have TATs. The other measure here is that high resolution is good. This is high resolution. This is lower resolution. You have much more points here, and means much more restraints. And when you have much more points and much more restraints, you need much more CPU, but the models are better. Okay? So high resolution is better, which means that experimentalists should do a lot of sequencing and put a lot of money in sequencing to get the highest resolution possible with the amount of money they have. The second thing is noise. We increase the amount of noise. And here again, distance RMSD versus what we call the TAT-bit correlation coefficient. This is an internal measure whether we are recapitulating the original matrix. This is not comparing the models, but this is comparing the contact map of the models with the original matrix. So it's an internal measure, and this first message is that there is a negative correlation, which is good because we know that when we get good TAT-bit SECs, it's likely to get good models. But then the noise level goes from yellow, little noise, to red, a lot of noise. The grade is in that direction, not in that direction, so which means it's pretty much insensitive. The quality of the final model is pretty much insensitive to the amount of noise. I have to say that the ones that have less noise, the correlation is a bit worse than when you have a lot of noise. So probably TAT-bit, which is based on a population data, is much better than when you have a variability in it than when you don't have any. There are other methods that are being available, like Rex3D and so on, that are probably better for single-cell data, I would say, than TAT-bit. Structural variability is not good. This is exactly the same map now, but you see the color goes from yellow to red in that direction, in the direction of the quality of the models in the y-axis. Which means that when you have a lot of structural variability in your original matrix, we are not able to build three-dimensional models that are reconstructing the original sets of variable structures. Okay? So we are quite sensitive to structural variability in that sense. But there is a good news here, is that we can try to figure it out before we actually build the three-dimensional models. We can figure it out whether the matrix is going to be a good matrix. It has a potentiality for building models. And the way we did that is by looking at properties of the matrix. This is, again, another one of the 67 matrices that we built. You see the TATs, more or less. These are just data. And we calculate 3 measures. One of them is the percentage of the significant contribution of the eigenvalues in the matrix. So you are looking for internal correlations in the matrix. If you have a lot of internal correlations in the matrix, that shows up here. In green here, in that area, is the actual contribution compared to a randomized matrix. What we do here is we randomize the points along the diagonal. We do not change the decay from the diagonal. We randomize the points, and then we get this orange line here. And these are the numbers that you get for the 67 matrices. This is the percentage of eigenvalues, and it has a negative correlation with the quality of the models. So the more contribution, the more green area here, the better the models are going to be at the end. The second measure that we look is at this distribution of the values here. Now you distribute the values. This is the frequency, and these are the z-score values of the matrix. And we calculate the skewness and kurtosis, which basically both of them are telling me how far from a normal distribution you are. Skewness, it tells you whether you are skewed towards negative or positive values, and the kurtosis tells you whether you have two peaks, one peak, a flat peak, etc., etc. And you can see that they also have very nice correlations to the quality of the models. There is also a very nice feature coming from the skewness, which is, it's a side effect, I think, that we're going to use. This is the same plot than before, but now I'm coloring here and making the dots proportional to the noise and the structural levels in the matrices. And if you look at all the points that have a lot of noise, this is a big blob, and they are yellow, which are these guys here, they are all negative skewness. They are all skewed towards negative values of the distributions. If you look at the structural variability with little noise, these are the little dots here, red dots, they are all positive distributions. So a priori, by looking only at the matrix, I can say whether we have a lot of structural variability or we have a lot of noise. And because I know structural variability is not good news for me, I can start picking up whether I'm going to be using that particular matrix for three-dimensional modeling. So I might save some CPU time here and expectations from the models. We went one step forward, and we built what we call the matrix modeling potential score. This is not a quality of the models, but a prediction based on the matrix data, whether the models are going to result in accurate models or not. And we just combine either the size, which is proportional, it's actually the resolution of the data, skewness, significant contribution of the eigenvectors, and kurtosis with these weights. And this is the correlation we get with the quality of the models, in this case, distance Spearman correlation coefficient. Very nice correlation, 0.84. So now, if I take one actual, this is actual data from the human high-C map, from Lieberman 8, no, in this case it's from our own group, François Ledilly did this work. So chromosome 1, this is about 8 megabases, few touch here, 1, 2, 3, 4 touch. This is the distribution, this is the size, and what you get at the end is an MMP score of 0.82, which means that I can go now and tell that this particular 0.82 MMP score with a accuracy of about 95% margin, we're going to get a distance Spearman correlation coefficient to the supposed to be structure of the genome of in between 0.7 and 0.9, which is pretty decent. So let me summarize what I'm saying here. TADs are good and high resolution is good. So if you are an experimentalist doing high C and you want to build three-dimensional models, put all your cash in sequencing. This is basically what that first message says. Noise is okay, so no need to worry too much whether you have false positive or false negative in your interaction data. The noise is canceled. Structural variability is not okay for three-dimensional model based on restraints, which means homogenize your cell population as much as possible, synchronize your cells, reduce the number of cells as much as possible. Instead of doing millions, do thousands of cells if you can, do the HICE experiment. And then additional values of this work is that we can differentiate between noise and structural variability, so we can go back to the experimentalist and tell them, use the matrix for something else than modeling, because we don't think it's yet at the level of accuracy that we need to build good models, and we can predict a priori the accuracy of the models. So let's thank really Marie, who did all the work here with the help of François and Davide, the rest of the group, and this was a nice collaboration with Ivan and the Serrano group at CLG. Thank you very much.
Questions for your approach? You talk about structural variability is not good, but could that be useful information by homogenizing the data? Are you getting rid of some useful information that you might calculate with single cell analysis?
Yeah, no, the data is good. The problem is that, so structural variability is not good for three-dimensional modeling based on restraints. because variability is good if we could capture it well. And I don't think we've yet captured it well, this variability. So if you really want, the message is if you really want to do three-dimensional modeling based on restraints in that type of approximation, which is a mean field force field restraint type of thing, if you really want to do it, try to homogenize as much as possible your data. Another thing I didn't mention is that the structure of variability that we put here is homogeneous across all the models. It's not that we have state A and the state B very clearly pinpoint and then we mix them. Each of the individual models that we put in the original matrix have a slightly different structure. So we still need to understand exactly what it means, homogeneous variability, structural variability, or a limited set of states in the matrix. That maybe those we could do much better than just homogeneous variability.
We will have